Hello, hi everyone, I'm Sandhya. In this video, we are going to study about meiosis, another cell division called as meiosis. Meiosis is also called as reductional division. Reductional division. This meiosis, this type of cell division mainly takes place in the reproductive cells, such that it results in the formation of a haploid set of chromosomes where the number of chromosomes are halved, reduced and matter. So that's why it's called as reductional division. So in sexual reproduction, the formation of gametes is very important, because sexual reproduction is nothing but the fusion of male and female gametes to produce a zygote. So the formation of the gametes, the formation of the haploid gametes takes place by this particular division called as meiosis. As this particular meiosis takes place in diploid cells and results in the formation of haploid cells, and the chromosome number is reduced from diploid to haploid. It is called as reductional division. So mainly this particular division takes place in diploid cells, and at the end of the meiosis, four haploid gametes are formed, or four haploid daughter cells are formed. Whereas in mitosis, two daughter cells are formed which are having same genetic constitution as the parents. But here, after meiosis, four haploid daughter cells are formed where the number of chromosomes is reduced to half. So in detail about this particular meiosis we are going to study. So this meiosis can be explained in two stages, meiosis 1 and meiosis 2. Okay, this meiosis 1 is mainly called as reductional division. Meiosis 2 is similar to that of mitosis. So meiosis 1, at the starting of the meiosis 1, at one stage only, the replication of the DNA takes place. Okay, that results in the formation of haploid gametes. Actually, this meiosis is divided into two, no? Meiosis 1 and meiosis 2. So in the beginning, here only the DNA replication takes place. One time the DNA replication takes place. As a result, by the end of these two, four will be present which are haploid. Then here in the meiosis 1, what happens? The pairing of the homologous chromosomes takes place. The crossing over takes place. Then crossing over takes place. That leads to the crossing over, and recombination takes place. So some changes are going to take place in meiosis 1. Then at the end of meiosis 2, four haploid cells are going to be formed. This is general characteristics of meiosis and matter. So this meiosis can be divided into two stages, meiosis 1, meiosis 2. Here prophase 1, metaphase 1, anaphase 1 and telophase 1. So here again, prophase 2, metaphase 2, anaphase 1, telophase 2, telophase 2, like that. Okay, so first we will study about the meiosis 1, which is the main reductional division. So in this meiosis 1, the prophase 1 is generally prolonged when compared to the prophase of mitosis. Okay, the prophase, if you take the prophase 1, it is somewhat prolonged with that when compared to that of the prophase of mitosis. This prophase 1 is again divided into 5 stages. First stage is leptotene stage. In leptotene stage, what happens? The compaction of the chromatograph takes place. That means condensation of the chromosomes is going to take place in leptotene. Coming on to zygote. Leptotene is formed by zygote stage. So in this stage, what is happening? Here in this stage, the pairing of the chromosomes is going to be taking place. That particular pairing is called as synapsis. So here in the leptotein, clearly we are going to see the chromosomes. Okay, in the zygotine, the pairing of the chromosomes takes place. That pairing is called as synapsis, and this synapsis between what chromosomes? Homologous chromosomes are going to get paired. Homologous chromosomes are going to get paired. That pairing is called as synapsis or synapsis. And because of the pairing, see like this, no? So what is happening here? Like this, because of the pairing, a complex is formed, which is nothing but a synaptonemal complex. Okay, and it results in the formation of a bivalent or tetrad. Okay, this bivalent or tetrad is clearly seen in the next stage called as pachytene. Okay, the next stage called as pachytene. Here what is happening? The bivalent and the tetrad stage of the chromosomes is clearly seen, and whenever the pairing is occurring at the sites, what is happening here? The crossing over is occurring. At the pairing junctions, and this crossing over is resulting in the formation of recombination nodules. As a result, here what is happening? Here? The recombination is occurring. And it is enzyme mediated. So here, which enzyme is responsible? The recombinase enzyme is responsible for the recombination because of the crossing over. The crossing over is going to take place at the synaptic complex. Okay, so and that crossing over is occurring between the homologous chromosomes. First, the pairing occurs. Pairing ke baad, after that, they develop into synaptonemal complex. And here at the junction, at the sides, the crossing over takes place, the recombination takes place. So what happens? Slowly these chromosomes are going to leave, but here they are going to still be linked together. Okay, linked together. Then after this, diplotein stage. What happens in the diplotein stage? The dissolution of the synaptonemal complex takes place. Dissolution of synaptonemal complex takes place. Synaptonemal complex takes place. As a result, they appear in the form of an X-shaped structure. They appear in the formation of an X-shaped structure like this. So this particular X-shaped structure is called as chiasmata. So chiasma can be seen in diplotein stage. Okay. Then coming on to diakinesis stage. Diakinesis stage is nothing but, in this stage, the terminalization means this is going to get dissociated. The terminalization of chiasmata takes place. Then the nuclear envelope is going to disappear. The nucleolus is going to disappear. Golgi complex, endoplasmic reticulum, they are going to disappear. And here terminalization of the chiasmata also is going to take place. Okay, so this is about the prophase 1. Moving on to metaphase 1 now. So here in the prophase 1, how are the chromosomes? Like this. Okay. Then here in metaphase, what happens? In metaphase 1, they are going to get aligned on the metaphasic plate or equatorial plate. Okay, and the spindle fibers are going to attach to these particular chromosomes. Then in anaphase 1, they are going to the centromere splits and they are going to move to the opposite poles. The chromosomes are going to move to the opposite poles because of the splitting of the centromere, and the spindle fibers are going to move these particular chromatids to the opposite poles like this. Then in the telophase, what happens? Telophase 1, the cluster of the chromosomes are going to form. They are going to develop into two daughter nuclei. So what happens here? Then after telophase, what happens? The karyokinesis has been taking place and cytokinesis has been taking place, and two daughter cells are formed here now. So after meiosis 1, two daughter cells are formed. Okay. So cluster of the chromosomes is going to taking place. The nuclear envelope develops. The Golgi complex nucleolus is going to develop, and after that, the cytokinesis takes place, resulting in the formation of two daughter cells. Now, these two daughter cells are going to enter into meiosis 2. They will enter into meiosis 2. This meiosis 2 is same as that of mitosis. Okay. So mitosis. Here at the beginning of the meiosis 1, the DNA replication takes place once. Okay. Here the DNA replication is not taking place. Okay. It will enter into the prophase 2. Okay. What happens here? The chromosomes can be clearly seen like this. Here also. Like this. Then after prophase 2, in prophase 2, what is happening? Like this, the chromosomes are formed. Okay, in prophase 2. Then in metaphase, phase 2, they are going to align at the equatorial plate with the spindle fibers like this. Like this. Then in anaphase 2, the centromere splits and they are going to move to the opposite poles. Like this. Then in telophase 2, the cluster of chromosomes are going to form. Then the nuclear envelope develops, the nucleolus develops, the Golgi complex ER and Golgi complex are going to develop. Then after this, the karyokinesis, then cytokinesis, which will be developing into 4 daughter haploid cells or haploid gametes. Okay, this is about the meiosis 2. Okay, like this. Then significance of meiosis. What is the significance of meiosis? Meiosis plays an important role. Meiosis plays an important role in the formation of gametes. The haploid gametes are formed because of the meiosis only. And because of this meiosis, the crossing over is taking place, the recombination is taking place, which is leading to the increase in the genetic variability, and genetic variability from one generation to another generation. And because of this crossing over and recombination, and because of this genetic variability, the variations are occurring, and variations are the reasons for the genetic evolution or the evolution purpose. So this meiosis plays an important role. When compared to mitosis, this meiosis character is an advanced one, and this plays a very, very important role in the formation of gametes and sexual reproduction. Thank you. So in meiosis, one must remember that prophase 1 of meiosis 1 plays a very, very, very important role. That is the phase where the actual genetic variability is taking place. Okay, and this prophase 1 is somewhat prolonged when compared to prophase of mitosis. And this particular prophase 1 is divided into leptotene, zygotene, pachytene, diplotene, and diakinesis. These are the 5 stages which are occurring in prophase 1. This is a small brush up. So in the leptotene, what is happening? Clearly we can see the chromosomes. But here we can see the nuclear envelope, then nucleolus we can see. But clearly the chromosomes are clearly seen here. In the zygote stage, what is happening? In the zygote stage, the homologous chromosomes are pairing up. Okay. Here, the homologous chromosomes are pairing up like this. That pairing is called as synapses. Okay. And those chromosomes which are getting paired up are homologous chromosomes. Okay. And because of this pairing, they're forming a complex structure which is called as synaptonemal complex. This synaptonemal complex, and they are developing into a bivalent or tetrad. It is not clearly seen in the zygotene stage, but in the pachytene stage, you can clearly see the bivalent and the tetrad stage. Okay, bivalent and tetrad stage. And at the sides of this particular complex, the crossing over takes place. The crossing over leads to recombination. Recombination is an enzyme mediated one. It takes place in the presence of an enzyme recombinase. And that is about the packet. Coming on to diplotene, what happens in the diplotene? The dissolution of the synaptonemal complex takes place. But at this side, at the crossing over side, they're still linked together, and results in the formation of an X-shaped structure which is called as chiasmata. Then when you put diakinesis is a state where the terminalization of the chiasmata takes place, and here completely the nuclear envelope and the nucleolus, Golgi complex, endoplasmic reticulum all are disappeared. Okay, disappearance of nuclear envelope, nucleolus, endoplasmic reticulum, and the Golgi complex takes place, and terminalization of the chiasmata takes place. And if it's an animal cell, the centrioles are co-duplicated into two anmata. Then if it's a plant cell, the cytoplasm is going to have the microtubules which are going to synthesize the spindle fibers. Then they get attached to this, and clearly in the metaphase 1, they are attached to the centromere and kinetochore, and those get aligned onto the equatorial plate. Then in anaphase, the centromere splits, and these particular chromatids are moved on to the opposite poles. In telophase 1, these chromosomes are going to get compacted, decondensation takes place, and they form into chromatin reticulum, and formation of nuclear envelope, nucleolus, Golgi complex, and endoplasmic reticulum are going to be seen. Then here we can clearly see the division of the nucleus. After that, cytokinesis takes place, which results in the formation of 2 cells. At the end of meiosis 1, it results in the formation of 2 cells. Then directly it will enter into the meiosis 2. And the stage between meiosis 1 and meiosis 2, it is also called as the gap between meiosis 1 and meiosis 2, it is called as it is also called as interkinesis. It is called as interkinesis. Okay, interkinesis. It is short-lived actually. It is short-lived. It is a stage between two meiotic divisions. It is short-lived, and it is followed by prophase 2 of the meiosis 2. So here this is same. Here also same resembles that of the normal mitosis. What happens in prophase 2? The dissolution of the nuclear envelope takes place. The compaction of the chromatography takes place, and the chromosomes are visible. Spindle fiber starts synthesizing. The Golgi complex, the nuclear envelope, nucleolus gets disappear. In metaphase 2, the spindle fibers are going to attach the centromere mainly to the kinetochore, and these chromosomes are going to get aligned on the metaphasic plate or the equatorial plate. But difference is that here two cells, it is going to occur simultaneously in these two cells. In anaphase, what happens? The centromere is going to get split up, and the chromatids will be moving on to the opposite poles. In telophase, these chromosomes are going to form into a cluster-like thing, and the decondensation takes place. They are going to form into chromatoreticulum. Then close to the nuclear envelope, the nucleolus is going to develop. The nucleolus is going to develop, and nucleus is going to develop. The nuclear envelope is going to develop. Golgi complex ER is going to develop. Then what happens after that? The cytokinesis takes place. The cell plate formation is going to take place. That results in the formation of middle lamella that divides the specular cell into two. So here two cells are there, no? So from one cell to another cell, two. Total 4 haploid cells are formed after meiosis 2.